Also, before I forget, please mute your cell phone. Okay, that is important. Thank you for coming to this small room. I was expecting actually more people, but this turns out to be the right size, similar to last time. Okay. By the way, if you don't know me, my name is Chuanmin Hu. I direct the optical oceanography lab, or Hu's lab, in this college. Okay. Anyway, welcome to the faculty show today. Before we begin, let me say a few words about the background of this show. Starting from last spring, on every Friday of the semester, first Friday, okay, we have a faculty seminar series as part of our in-reach and outreach activities. So the purpose is to provide a forum for you, especially the new students, to get a better idea of what they do, to learn from their expertise, to share their excitement, to develop possible collaborations. Okay? I know you may get some information about themselves and their labs from the web. Okay, from their introduction page. But this is where you can meet the people, okay, and get more interactions. Okay. So for that, I would like to ask you to encourage your classmates, friends, to come to this seminar series next time. If for some reason, for example, they're at sea or have meetings, they can't come this time. So this is an important event. Let's see. After the recent retirement of Ted Van Vliet, Jose Torres, and David Mann, we have about 24 or 25 professors. But we're going to have 3 new professors joining us next spring or summer. So we have 27, 28, plus our dean, Jackie Dixon. So we have 28 people. So every seminar series, we have 8, more or less, 8 people giving their talks. So in about 3 to 4 semesters, we have a cycle. Then we start all over again. Okay? So that's about the background. Any questions? If no, so let's begin with our professor, Muller Karger. Okay. Let me pass you the microphone.
I'm not going to take my shirt off. Well, good morning, everybody, and thanks for roping me into this, Chuanmin. So my name is Muller Karger. I'm a professor here at the College of Marine Science. I've been here since 1988, and I was brought here by Dr. John Walsh. He brought me from the University of Maryland as a postdoc, and then I graduated to be a professor. So over these years, long time, 25 years or so, I've been developing a lab, and this lab has grown in many different directions. It's a pretty eclectic lab, and it really, all that happens there is because of the people that we bring into the lab. So we have worked in fisheries and red tides and water quality, looking at coral reefs, and we maintain a time series in the Caribbean called the CARIACO time series. And we've also collect satellite data with these antennas, one of them that is outside here in the parking lot, another one on top of the building. So I'll very briefly talk about some of these things, very superficially, really, because there's not time to get into any one of these things. So in our lab, what we really strive to do is to try to understand how the world changed over time, how it changes in space and in time, and to educate people, the public as well as new scientists, and how to use data, what what observations about the world mean and how you use them. So right at the moment, and this is kind of pretty much for the past 20 years, the lab has had about 16 people, including myself. Right now we have 5 graduate students, and 4 of them are PhDs. We just graduated a few over the past year. Those have stayed on as postdocs, so we have now 5 postdocs, which is kind of a little crazy. One of them is in Puerto Rico. We have 2 systems engineers, 3 research associates. Basically, they work as my right, so I have several right hands. And we have an intern from USF St. Pete. And we often have visitors coming and staying in our lab from different parts of the world. This is the team. It's hard to get them all together in one time to get a picture. These are the antennas that we have. These are things like, we call this an X-band antenna, and we use that to collect data from 2 satellites that NASA flies, and especially we focus on a sensor called MODIS. There's 2 MODISes on these 2 different satellites. And the other one is on top of the FWRI building. That's an L-band antenna. And it used to collect C waves also, so we collected C waves for many years. Now we collect data from something called the AVHRR, which we use to look at temperature of the surface ocean. This one is used to collect the MODIS data, gives us temperature as well as ocean color, the color of the. So with these antennas, we can see from the Southern Caribbean all the way to the Great Lakes in Canada. So it covers quite a bit of real estate, and we collect many images per day. All of those images then are put on the web very quickly within a few minutes. And the advantage of these antennas is that you don't have to wait 8 hours or 10 hours for the data to be available at NASA or at NOAA. You get them within minutes. So these are some of the images that we get. These are just examples. We share them with everybody openly. We share them with the public, with other scientists. We share them with these regional associations of the Integrated Ocean Observing System that we call GICUS and SECURA. We give them to private companies, and they make business with that, like ROFs. So this is an ocean color example. This is what you would see if you were out on a space station. So it's sort of what we call that red, green, blue type of true color images and some temperature images. So we use these data to do measurements. So they're not just pretty pictures. You see things moving around. You see things changing over the seasons. And we now have literally some 20 years or so of data, in this case of 1 kilometer resolution, both for temperature and ocean color. So in this case, we're working with NOAA. This is Maria Vega Rodriguez. She's working in a project jointly with NASA Ames and with NOAA, the NOAA Coral Reef Watch program. And we have brought the Coral Reef Watch program products, which tend to be about 50 kilometers per pixel, so something like of this nature. And we merge it with our data at 1 kilometer to try to estimate the stress that coral reef populations may be experiencing due to temperature. So this kind of measures how many days above a certain threshold of temperature coral reefs may be experiencing any one year. And then we compare that, and we work with FWRI to look at the species composition on the bottom, on how coral reefs may have changed over time, whether the coverage of coral reefs has been decreasing or whether their species composition has been decreasing, how that's related to these environmental parameters. Another thing that we've done is this product that we call the Millennium Coral Reef Map. The Millennium Coral Reef Map is the first, and so far the only, global coral reef map at 30-meter resolution, and we did that with Landsat data. We're now gearing up to do it. Landsat data is 30-meter resolution per pixel, so we're now gearing up to do this at 2-meter resolution with a global data set that is produced by a satellite called Worldview 2. So we have the data. It's a major, huge project, so we're going to see how we actually accomplish that. It took us many years to do Millennium with Landsat, and I don't know how we're going to do this at 2-meter resolution, but we're going to try. Enya Soto. Enya just graduated. She's got a postdoc offer in Brazil. She worked with Chuanmin and myself looking at red tides, but the interesting thing is that she tried to compare what we see here in the Florida red tides with what we see in Mexico. And so she found that there are some differences in the species composition that you can actually tell by satellite. And so she spent a lot of effort. She collected some of the first optical measurements in Mexican waters in the Gulf of Mexico. So quite a job, and hopefully we can continue this work even if she's remote in Brazil. Sanai is a PhD student with us who works with NOAA very, very closely on their CMAP data. Sanai uses several different satellite products from ocean color, temperature, sea surface elevation, which is kind of contoured here over these images, to try to understand the distribution of pelagic fish larvae. NOAA goes out every year, they've been going out every year since the mid-'80s, in the spring to collect larvae for pelagic fish, especially the whole purpose, really, the key purpose here is to try to understand the distribution of larvae of bluefin tuna. So there's other types of tunas and other pelagic. So Sanai is going through the data and trying to understand if these things are related, the distribution of these larvae are related to fronts or some other features in the Gulf. Brendan O'Connor, he also graduated as a master's. He used MODIS satellite data to look at the fluorescence of phytoplankton in the Gulf of Mexico, again working with Chuanmin and myself, to understand the patterns of phytoplankton distribution in the northern Gulf of Mexico and how that may have been affected by the Deepwater Horizon oil spill. He also worked some, and didn't go very deep into it, into these data. These data are collected by an airplane that NASA flew. It's a version of the U-2 spy plane, but it's a research aircraft, and they fly a hyperspectral camera, and they did that over that whole region during the Deepwater Horizon oil spill to try to understand how far the spill may have gone. So Brendan worked on these data trying to identify features in the spectrum. This is color here, or wavelength, and intensity of light that you would measure from the sensor, and you see some features that are there when there's oil, and you don't see these features when there is no oil. So he was trying to understand if you could use these images kind of in an automated way to detect oil. Matt McCarthy and Bob Chen, Dan Otis, sitting back here, they're working on an EPA project to look at water quality using satellites. So the idea here is to try to complement some of the field measurements that water quality managers make. They make them typically once a month or not even that often. With the satellites, we can see the whole estuary within a minute or so, and we can see them frequently, often several times a week if there's no clouds. But we're not trying to do just Tampa Bay, we're trying to do all the estuaries in the northern Gulf of Mexico. We're getting into Mexico, and we included Puerto Rico. So that's a big project. Matt is looking at land use change over the past 20 years and see if that has an impact on water quality. So he's using these maps of land use that have been produced by the government over several epochs and see if that has an impact. Gerald Tow, he's a postdoc in the lab, and he's an expert in looking at water optical properties. So he's put together these devices that you can lower in the water, or you can take around with divers and measure the color of coral reefs, measure the color of the bottom, and then try to understand what it is that you see from above the surface, from a boat, or from a satellite. So this was part of an experiment that we did over the past year with NASA using unmanned aerial aircraft over the Florida Keys. And CJ Reynolds and Abdiel Laureano. Abdiel is a new student. He just arrived this semester. CJ is a research associate. They're working on a project that we just got funded by the National Science Foundation as part of an international program that the Belmont Forum funded. So the Belmont Forum is moving towards something called Future Earth, and so they're trying to marry the natural and the social sciences, and they're trying to fund these types of programs where you're trying to help inform science what people, the different communities need, and then try to have the scientists co-develop or co-design solutions for these communities. So we're working with people in England and people in Brazil, some group in Maine, to try to understand some of these vulnerabilities in coastal communities in these 3 countries. One other thing that we do every other year or so, and we've been doing it since about 2001, is we have a class in ocean policy. And so this is really a work of the students of the college, where they go through and try to understand how this country developed the policies that we have right now in place regarding the ocean and regarding the coast. And then they go through a process of engaging the media. They go up to Tallahassee for Florida Oceans Day, and then they go to Washington, D.C., and they engage with Congress people. They try to talk to people that are part of the White House or different agencies of the executive. They talk to staffers. They talk to non-governmental agencies. So I think it's quite an experience for people in this college that very, very few other marine science colleges have. So I'm very proud of that, and I think the students get a lot of it, a lot out of it. So in terms of the CARIACO time series, this is one project I'm going to focus a little bit more on. Several professors here, Bob Weisberg, John, Dave Hollander, have been involved in this over the time. This project is now 18 years in work. We just got funded for another cycle, so we're going to continue. And the work here is in this basin in the Southern Caribbean. So the point of this is a deep basin. It's on the continental shelf. It's kind of a hole in the continental shelf, and it's open to the Caribbean in the upper 140 meters, and then it's basically quiescent and enclosed deeper than that. So it's an area where you can see a lot of upwelling because the trade winds race right past the coast, but it's very seasonal. So the trade winds intensify during part of the year, and the other half of the year you have basically a rainy season. So during the upwelling season, typically between about January and May, you have this upwelling stimulates phytoplankton growth, and you have plumes of high concentrations of phytoplankton stretching this way. And you also have rivers. This is the Orinoco River. It kind of looks the same, and that's an artifact in the images. The Orinoco, for example, doesn't go into CARIACO. We found that out. But we do have some local rivers that impact the basin. And so you have alternation between biomass from phytoplankton at one time of the year and then river input during the other half of the year. And both of those things create particles that sink to the bottom. So as the stuff sinks to the bottom, and because this basin is enclosed, the waters become anoxic. The stuff just rots, and there's lots of bacteria growth, and these bacteria consume this material, and you run out of oxygen. So it's an incredible anoxic basin from about 250 meters to about 1,400 meters. So the point, and the key in this project are Laura Lorenzoni and Enrique Montes. Many of you know them. The point here is that we have a time series that sort of a basic operational objective is to have a platform for people to collect samples over long periods of time and compare things, put their measurements in the context of a time series. The scientific goals are to understand the linkages between what we see at the surface, especially the production of particles or whatever comes in through rivers, and whatever lands on the bottom. And the importance of this is that CARIACO is a place where historically there's been paleoclimate studies it's very visible because the paleoclimate studies have used the CARIACO basin as a place where the chronology, these sediments settle on the bottom, and there's no organisms, no macrofauna to rework the sediment. So the layers of sediment are very fine and they record paleoclimate very well. So these sediments have been used by a lot of people to basically build a chronology to calibrate other paleo records like Iceland ice cores, for example, or Greenland ice cores. So the project is it involves a lot of people. We coordinate the project from here. We collect some of the measurements locally. We do all the satellite data, for example. We we coordinate with all these other people what kind of measurements we're going to do. We collect data for the University of Miami. They do our dissolved organic carbon. The Stony Brook University, Mary Scranton and Gordon Taylor do bacteria and bacteria productivity studies. Bob Fennell at the University of South Carolina does all the sediment work, and so we have collaborations with at least 5 universities in Venezuela where each one contributes a piece of the puzzle. We maintain a mooring there. The mooring has sediment traps at one point for 6 years, I think. Bob Weisberg had current meters on this mooring. And we collect an enormous amount of variables every month, and we've done that for 18 years. Some months we go out to up to 3 times per month. So it's a tremendous effort, and it's built an enormous data set. So what's happened in the Caribbean since we started back in 1995, is we've seen a gradual increase in the temperature. And so the question is why? And is that coherent? Does that represent what's happening over the Atlantic, for example? So is this place representative of a larger area? So we find, yeah, this increase in temperature over the Atlantic, or the tropical Atlantic anyway, is real. I mean, we've seen about a 1 degree increase in the past 18 years. And a lot of that has been because the trade winds have at least decreased in a relative sense over this period. So what we see is that the water is warming, the mixed layer is shallower. We have less chlorophyll every year at the surface compared to what we used to see. This is month here, so this is basically the upwelling season. This is the quiescence season. So we have less phytoplankton growth. And we're seeing other changes. I mean, so for example, the sardine fishery collapsed in the southern Caribbean almost completely. And that happened in this time frame here. So something seemed to be correlated with higher trophic levels in ways that we can talk about and explore trophic cascades and things like that. Another thing that we see, so this is the phytoplankton. Phytoplankton concentration not only decreased, but the composition of the phytoplankton changed, where we had larger cells, more diatoms early in the series, and then smaller cells later on. What we see also is that CO2 is increasing, so even though this is a continental margin upwelling zone where you would bring CO2 from deep water and upwell it near the coast, so basically you're bringing subtropical underwater and bringing it to the surface, we do see an increase in the concentration of CO2 in surface waters in this area. So that probably has several reasons. One is it's not just connected to an increase of CO2 in the atmosphere. The water is warming, so as the water warms, you would see an increase in the partial pressure. Phytoplankton are taking up less because there's fewer phytoplankton, so you don't have that effect of the phytoplankton taking away the CO2, so that would also lead to an. So this signal is not something easy to interpret, and so part of our effort is trying to understand why we see this, because not simply CO2 is increasing in the air. There's much more complex story. So I'm going to stop here, but this is kind of a very, very brief thumbnail sketch of what happens in my lab, and it wouldn't happen if it weren't for the people in the lab. So I really I owe everything to my team. So thank you.